Hello everyone. Today our study topic is transcription. And to study transcription, first we will see what is central dogma. Gene expression is called central dogma, and there are two steps of gene expression: transcription and translation. In the transcription, RNA will be synthesized from DNA, and in translation, protein will be synthesized from RNA. From the double helical DNA, first messenger RNA will be synthesized and then protein will be synthesized. Now come towards our main topic, transcription. To make RNA, we need the DNA, and from that DNA, gene sequence will be copied. And it is the first step of gene expression. And to synthesize RNA polymerase enzyme is required. Main process of transcription is the same in the prokaryote transcription and in eukaryote transcription, but there are slight differences which we will study side by side in this topic. In the eukaryotes, there are three type of RNA polymerases. RNA polymerase one will synthesize ribosomal RNA, and RNA polymerase 2 will synthesize messenger RNA, and RNA polymerase 3 will synthesize transfer RNA. However, in the prokaryotes, there is only one type of RNA polymerase which will synthesize all the three types of RNA: messenger RNA, ribosomal RNA, and transfer RNA. In the DNA, this is the double helical DNA, and we have two strands of the DNA. One is the coding strand or sense strand, which will not be used in the transcription, and the other strand is the template strand or antisense strand, and that strand is called antisense strand because this strand will not synthesize directly protein. First it will synthesize messenger RNA and then it will synthesize protein, and that template strand will be used in the transcription process to synthesize RNA. Now on that DNA region, there is a specific on that DNA, there's a specific region on which the DNA can be transcribed, on which transcription can occur. So that region is called promoter region. That region is called promoter region, where RNA polymerase will be bind and will start transcription process. In the eukaryotes and prokaryotes, these sites are different. In the eukaryotes, the sequence TTGACA or minus 35 sequence and TATAAT or minus 10 sequence will be used for the promoter region. However, in the prokaryotes, the promoter region in TTGACA minus 75 and TATAAT minus 25 sequence. What is actually this sequence? In the promoter region, there will be that sequence on which RNA polymerase will be bind, and minus 35 and minus 10 means that where the genes expression will be gene transcription will start. Before that region, there will be a sequence before that region, and there is a numbering of the sequence. Minus 35 is the number of that sequence. It is minus because it is upstream to that region. Upstream means that it is before that region. If it would be the downstream, then it should be plus, but here in the transcription process, upstream region is used, so it is minus 35, and 35 is the number, and that is the sequence on which RNA polymerase will bind, and then after that gene will be copied. RNA polymerase is composed of two factors: sigma factor and coenzyme. Sigma factor is actually that part of RNA polymerase which will just initiate the transcription, and it will correctly position the RNA polymerase to the promoter site. After uh, correctly positioning the RNA polymerase on the promoter and after initiating the transcription, sigma factor will release from the RNA polymerase. And the remaining part of the RNA polymerase is the core enzyme, which will perform the main function. It will copy the DNA sequence to make the RNA, and it will move RNA polymerase enzyme on the template strand. When the RNA polymerase will bind to the promoter site, there will form a transcription bubble. To form the transcription bubble, there are some other proteins which are also involved, and first we'll see what is the transcription bubble. When the two strands move apart by uncoiling and unwinding of the duplex to form bubble with the help of RNA polymerase. This bubble is called transcription bubble. Above is the coding strand. This is the template strand. Here RNA polymerase will bind on the promoter site, and there are some proteins involved like helicase protein, which will unwind these two double-stranded DNA and produce the bubble. And there are also some other proteins, single stranded binding proteins, which will bind to that uh, template strand and the coding strand, so that these uh, two strands stay apart from each other. So this is the transcription bubble. Now, how transcription occurs? After binding the RNA polymerase and initiation of the transcription, mm, RNA polymerase will start making the copy of the DNA, and it will be synthesized in the 5' to 3' direction. As it will move in the 5' to 3' direction, so the template strand will be 3' to 5' strand, and the newly synthesized strand will be 5' to 3' in direction. So RNA synthesis mm, will continue until it reaches the termination point. And when termination will be done, when it will receive the stop signal, which is the GC sequence. Termination is actually a GC hairpin structure. GC sequence is followed by the AT and four or more polyuribonucleotides followed by GC. There will be a series of AT sequence and there will be a GC sequence, and this hairpin structure will be formed. Then after the GC sequence, U-ribonucleotide will be followed. And this when the newly synthesized strand of RNA will get this sequence, then it will stop more synthesis of RNA. This is the stop signal for RNA synthesis. When it will get this sequence, then the more RNA synthesis will be stopped. Then after the transcription process completed, after the RNA synthesis completed, there is some modification to that synthesized RNA, because the newly synthesized RNA will have to be protected from some enzymes. But in the prokaryotes and eukaryotes, there is a slight difference. In the prokaryotes, as the messenger RNA, other RNA which is synthesized is directly released into the cytoplasm, because it is also synthesized in the cytoplasm, and there is no nucleus in the prokaryotes, and their DNA region is present in the DNA is present in the cytoplasm. It is directly submerged in the cytoplasm, so RNA of the prokaryotes, there is no need of further modification because transcription and translation both are being done in the same cytoplasm. However, in the eukaryotes, messenger RNA moves from the nucleus to the cytoplasm, so we have to protect it from the several type of enzymes like nucleases and phosphatases, which can destroy the RNA. So on the 5 prime side of the RNA, which is also called the head, there 7 methyl GTP guanine triphosphate will be attached, and on that 3 prime side, which is also called tail side, polyadenylate sequence will be attached. When this sequence will be attached on the both sides, then no phosphatases and no nucleases can destroy this synthesized RNA. So here we are done with our topic, and if you have any question, you can comment me and you can ask. And if you like my video, if you like my topic, then please like, share and subscribe. Thank you.